The introduction, and thank you to James Chong and Jay for the invitation and for bringing this meeting to Australia. It's great to be here and to be able to share our work with you. So today I'm going to talk to you about the work we've been doing on trying to understand the regenerative capacity of the neonatal heart, and in particular to tease apart some of the underlying molecular mechanisms. So in contrast to the extremely limited regenerative capacity of the adult heart, the neonatal heart retains a transient capacity for cardiac regeneration, which is lost shortly after birth. We've demonstrated that in neonatal mice, and more recent studies from others have demonstrated that a similar phenomenon occurs in neonatal pigs, that the heart is able to regenerate following injury at postnatal day 1, and then this regenerative capacity sharply declines during the first week of postnatal life, such that the cardiac injury response at later stages of development becomes characterised by a lack of regeneration and proliferation of cardiomyocytes and fibrotic wound healing. So really the key drivers for much of our research over the last few years have been in trying to understand what drives heart regeneration in the neonate, and secondly, what are the barriers to heart regeneration in adults? And so to try and understand these mechanisms, we set about undertaking a large transcriptional analysis of the neonatal and adult heart before and after myocardial infarction. And this work was driven by a PhD student in the lab, Greg Quaife-Ryan, who's here at this meeting, and I encourage you to go and talk to Greg about this paper. And essentially what Greg did is he took neonatal mice postnatal day 1 that had been subjected to either myocardial infarction or sham surgery, and adult mice that had been similarly subjected to either LED ligation or sham surgery. And he then purified the major cardiac cell populations in the heart, including cardiomyocytes, CD31 positive endothelial cells, CD90 positive, CD31 negative, CD45 negative fibroblasts, and CD45 positive leukocyte population. And he subjected all of these cardiac cell populations to RNA sequencing in the neonatal and adult heart with and without myocardial infarction. And the goal was really to map the transcriptional networks that underpin postnatal cardiac development and maturation, as well as that distinguish both the reparative and regenerative responses of the mammalian heart following injury. And so we recently published this transcriptional resource and have made this data set available to the scientific community. And essentially, we've found that there are indeed major transcriptional shifts in all of the major cardiac cell types in the heart during this neonatal to adult transition. That is, all of the cardiac cell types, fibroblasts, endothelial cells, myocytes, and leukocytes, undergo major transcriptional changes as the heart matures from neonatal to adult stages of development. And one of the key findings from this data set is summarised here in this principal component analysis, where we found that there was a striking reversion towards a neonatal transcriptional signature in the adult fibroblasts and leukocyte populations following myocardial infarction. That is, following cardiac injury in adulthood, there is a large-scale reactivation of the neonatal transcriptional program in both the fibroblasts and the leukocyte populations. However, in contrast, cardiomyocytes and to a lesser extent endothelial cells fail to reactivate this neonatal gene program following cardiac injury in adulthood. So what are these transcriptional networks that fail to become reactivated or re-engaged in the adult cardiomyocyte following myocardial infarction? When we drill down in a bit more detail into these transcriptional networks that are shut down in all of the major cardiac cell populations postnatally, reactivated in fibroblasts and leukocytes following myocardial infarction, but that fail to become reactivated in the myocyte and endothelial cell populations, we find that these transcriptional networks are highly enriched for transcription factors, pathways and gene ontology terms related to cell cycle, mitosis and various aspects of cell cycle control. So adult cardiomyocytes and also endothelial cells, by and large, fail to reactivate this cell cycle transcriptional network following myocardial infarction. So we next wanted to understand what might be some of the key upstream signalling pathways that govern this cell cycle transcriptional network. And so we've gone back to our transcriptional data sets and we've looked at the major signalling pathways that appear to drive these neonatal transcriptional networks. And what we found is that around 50% of the transcripts in the regenerative neonatal cardiomyocytes are associated with components of the Wnt beta-catenin signalling pathway. And so we've subsequently performed additional characterization of this pathway during postnatal heart development. And what we found is that Wnt beta-catenin activity sharply declines during the first postnatal week of development in the mouse. And this is shown in the top panel here. This is immunofluorescence staining for a nuclear localized active form of beta-catenin. You can see that many cardiomyocytes at postnatal day 1 express this active form of beta-catenin in the nucleus. By postnatal day 7, there are only a few cardiomyocytes and virtually no cardiomyocytes after postnatal day 14. Similarly, using a top flash luciferase reporter, which reports on TCF transcriptional activity, you can see that there is a sharp decline in beta-catenin TCF transcriptional activity after the first postnatal week of development. So is Wnt beta-catenin signaling a major regulator of cardiomyocyte proliferation? Well, many studies have shown that activating this signaling pathway is a potent stimulus for proliferation in immature cardiomyocytes. And we've confirmed these findings. Here you're looking at iPSC-derived cardiomyocytes in vitro, and if we activate Wnt beta-catenin signaling using the GSK3 inhibitor Kaya, we see an increase in the percentage of mitotic cardiomyocytes, and similarly, constitutively active form of beta-catenin results also induces cardiomyocyte mitosis to a similar extent to constitutively active YAP, which is one of the most well-characterized and potent inducers of cardiomyocyte proliferation. So if we go in vivo and we activate beta-catenin in neonatal mice using adeno-associated viral delivery of a constitutively active form of beta-catenin, we similarly see that in the neonatal setting, constitutively active beta-catenin is able to promote these immature cardiomyocytes to go into cycle. So what are the target genes regulated by beta-catenin in immature proliferative cardiomyocytes? And so to look at this, we've performed ChIP-seq analysis for TCF7L2 and cross-referenced these data in iPSC-derived cardiomyocytes to RNA sequencing data in both human iPSC-derived cardiomyocytes as well as the mouse. And we found that many of these TCF7L2 target genes are enriched for genes involved in cell division, DNA replication, and various aspects of cell cycle control. So beta-catenin targets many genes involved in proliferation in immature, highly proliferative cardiomyocytes, and we found that these motifs are highly enriched not only for TCF7, but also for TEAD3, which is a major transcription factor that partners with YAP, downstream regulator of the hippo signaling pathway. So we next wanted to determine whether beta-catenin was sufficient to drive adult cardiomyocytes back into cycle. And so for these experiments, we've taken adult mice at 8 weeks of age. We subjected them to myocardial infarction by permanent occlusion of the LAD, and we then injected AAVs expressing either a controlled GFP, constitutively active beta-catenin, or a constitutively active form of YAP around the infarct border zone, and we tracked these mice by serial echocardiography out to 4 weeks post-infarction, and then performed histology and RNA sequencing. And so what you can see here is that both constitutively active YAP as well as constitutively active beta-catenin had a cardioprotective effect and improved cardiac function, both ejection fraction and fractional shortening, following myocardial infarction in adult mice. And this was associated with a reduction in cardiac fibrosis in the mice treated with constitutively active beta-catenin. However, beta-catenin failed to reactivate or induce cell cycle re-entry in adult cardiomyocytes. And this was in contrast to constitutively active YAP, which induced a significant, albeit small degree of adult cardiomyocyte proliferation. So why does beta-catenin drive such a potent proliferative response in immature cardiomyocytes, but fail to reactivate proliferation and drive these cell cycle processes in adult cardiomyocytes? And we've tried to probe some of these underlying mechanisms by taking these adult mouse hearts, isolating and purifying the cardiomyocyte fraction, and then performing RNA sequencing on the hearts that were treated with either the controlled GFP or the constitutively active beta-catenin. And what we found was that in contrast to the immature, highly proliferative cardiomyocytes, beta-catenin failed to activate those cell cycle transcriptional networks, and instead it regulated a suite of genes involved in inflammation and metabolism. And in particular, there was strong regulation of these two beta-catenin target genes, KLF15 and PPAR-gamma, which are both involved in cardiomyocyte metabolism. So why do adult cardiomyocytes fail to engage this neonatal proliferative program? We've turned our attention to chromatin accessibility as one potential mechanism by which these transcriptional networks are unable to be re-engaged in the adult setting. And we recently published this work, which was performed by another PhD student, Evangeline Sim, in the lab, whereby we purified cardiomyocyte nuclei from mouse hearts at postnatal day 1, postnatal day 14, and postnatal day 56, and we subjected these to RNA sequencing or ATAC-seq to look at open chromatin regions across the genome. And what we found was that there was indeed profound remodeling of chromatin architecture during postnatal development in the mouse, whereby there were major shifts in chromatin accessibility around genes involved in cell cycle control and cytoskeletal rearrangement and metabolism. And when we integrated these data sets, we found that there was a loss of chromatin accessibility around cell cycle transcription factor target genes during this postnatal maturation process. And we believe this might be one of the reasons why adult cardiomyocytes are unable to re-engage these developmental transcriptional programs during postnatal maturation. So what are the upstream drivers of this cardiomyocyte maturation process? And I want to end this talk by discussing some of the potential mechanisms that we think play a major role in shutting down cardiomyocyte proliferative capacity after birth. And this work has been an ongoing and long-term collaboration with James Hudson when we were both at the University of Queensland, and subsequently since we both left the University of Queensland. And this work really builds on major advances in the field in our ability to generate cardiomyocytes from human pluripotent stem cells. And we've heard a lot at this meeting about how we're using these pluripotent stem cell-derived cardiomyocytes for cardiac tissue engineering and regenerative medicine. And you're going to hear more from James later in the meeting about how we're using these platforms for drug screening. But what I want to talk about today is how we can also use these model systems to better understand cardiac development and in particular to identify drivers of cardiomyocyte maturation. And so these studies were driven by a postdoc in the lab, Richard Mills, and also by a PhD student, Holly Voges. And so the platform that we've used was recently published, and we have generated 3D cardiac organoids from pluripotent stem cells, which are cultured in a 96-well device. These tissues are spontaneously contractile, they're force-producing, and we can, because they're cultured in this 96-well platform, we can use them for high-throughput screening applications. And so we undertook a screen for potential regulators of cardiomyocyte maturation using this pluripotent stem cell-derived cardiac organoid platform. And the way we defined maturation in our screen was looking for factors that increase the force of contraction of the tissues, that increase the MLC2V expression, which is a maturation marker of ventricular cardiomyocytes, and that also decrease cell cycle activity as assessed by Ki67 staining. And we screen many factors, including extracellular matrix components, growth factors, hormones, and metabolic substrates. This work was recently published, so I won't go into detail regarding all of the factors that were screened, but this required characterization of a large number of tissues, which was only possible because of the screening capabilities of this platform. And to cut a long story short, what we found was that a major driver of cardiomyocyte maturation and cardiomyocyte cell cycle arrest was a metabolic switch from glycolysis to fatty acid oxidation. So if we force cardiomyocytes to use fatty acids rather than glucose, this pulled them out of cycle, reduced the cardiomyocyte mitosis rates to levels that are seen in the postnatal hearts, postnatal human heart. It reduced Ki67 positive cardiomyocytes, and not only did it decrease cell cycle activity, but these cardiomyocytes under these maturation conditions also became refractory to developmental mitogens, including the GSK3 inhibitor Kia. So essentially, this recapitulates a key step in postnatal cardiac development when the myocytes not only withdraw from the cell cycle, but they become refractory to mitogens. So what do we think might be going on? Our current working model is that the postnatal switch to highly oxidative environment, which is associated with major shifts in metabolic substrate availability, results in a switch from glycolysis to fatty acid oxidation after birth. And we believe that this switch in metabolism plays a key role in shutting down the cell cycle in mammalian cardiomyocytes. We've shown that in part this operates through transcriptional shutdown of both beta-catenin and YAP target genes. And we are broadly interested in the role of metabolism in cardiomyocyte transcriptional regulation. We believe this is a largely unexplored and important area for future research. So I'd like to end there. I think I've acknowledged all of the key people as I've gone through the talk, and I'm happy to take questions. Thank you.
So I think we'll take one quick question, and if there's any further questions, we can extend it into the lunch break.
That's a great talk. So any particular, more comments on what type of metabolites that would really cause such a fundamental chromatin remodeling?
So there's been a lot of work done looking at the pentose phosphate pathway and branching pathways of the glycolysis metabolic pathways, including 1-carbon metabolism, that are important for generating the building blocks and nucleotides required for DNA synthesis. So we actually require glycolysis for DNA synthesis because that's where nucleotides are generated from. In terms of chromatin remodeling, there's been a lot of work looking at 1-carbon metabolism, which is required for production of methyl groups, required for DNA and histone modifications, and also the TCA cycle feeds into protein and histone acetylation as well. So there are many links between the core metabolic pathways and epigenetic regulation. This has been looked at a lot in the cancer field, but I think it's something that we need to think about more in the cardiac field in terms of transcriptional regulation at a global scale.
All right, thank you, Enzo.